So it is a pleasure for us to have today with us Professor Nitzan Censor from the School of Psychology in Tel Aviv University. Professor Censor received his master's in physics and a PhD in neuroscience from the Weizmann Institute of Science, studying perceptual learning under the supervision of Professor Dov Sagi. He then completed his postdoc training at NIH at Professor Leonardo Cohen's lab, studying motor learning and memory. His lab at Tel Aviv University investigates the mechanisms by which skill learning and memory are modulated over time with substantiated breakthroughs using a multimodal approach combining behavioral psychophysics, non-invasive brain stimulation, and neuroimaging. He has dozens of publications, many of which are published in leading journals as Nature Neuroscience, Nature Neuroscience Reviews, Current Biology, Neuron, Trends in Cognitive Neuroscience, and more. He has received multiple grants and awards, including recently the prestigious ERC Consolidator grant. His work is often covered by the media. So without further ado, welcome Nitzan to our seminar, and thanks for joining us to share your research with us today.
Toda Sharon, thanks. It's for the kind introduction, and it's my pleasure to be here virtually. I'm going to talk about a novel learning mechanism in the human brain. We started from perceptual learning, but now we've expanded our work to additional learning and memory domains. So I will start with perceptual learning, and the classical view of perceptual learning, which is improvement in perceptual thresholds through repeated practice. It started more than 3 decades ago in studies in various tasks, which have showed that human perception can be improved through repeated practice. Even in the mature adult human brain, challenging the existing dogma until then of the temporal time window of plasticity. How were these improvements, or are these improvements usually obtained? Well, through repeated practice with hundreds and thousands of exposures to visual stimuli. Here you can see the schematics of a standard learning curve. You can see the perceptual threshold on the y-axis, and it very gradually decreases with repeated practice. We asked the following: is this the only route for inducing plasticity in the visual system which enables perceptual learning, or are there more economic ways to achieve these improvements? And our rationale was inspired by frameworks in perceptual learning, showing that more is not necessarily better in terms of practice, and studies from additional memory domains under the framework of memory reactivation, showing that often once an existing memory is reactivated, then there opens up a time window in which this memory can be modulated. It could be degraded, but also strengthened. And I will expand on that in the next few slides. But just to finish the general rationale leading to these first set of studies, then with this rationale in mind, what we asked is whether we can incorporate these brief exposures serving as reactivation trials into the field of perceptual learning, replacing these hundreds and thousands of training trials with only brief exposures to the learned information. Before I continue with these lines of studies, I will first expand a bit on the basic inspiration to use this reactivation framework. So I will talk here a bit about consolidation, memory consolidation in general. And according to the classic model, as you see here, so Y would be the the y-axis would be the memory strength, and the x-axis some arbitrary time scale. So once the brain is exposed to information, then this information is stored in a short-term memory. And by definition, this short-term memory, the strength of the memory decreases very rapidly, and that's why it's termed short-term memory. However, with efficient consolidation, the memory is transformed from a short-term memory to a long-term memory, as you can see here. And then it's stored as a long-term memory trace in the brain. There may be some gradual process of a long-term forgetting, but in general, it's stored as a long-term memory trace. And the important point to remember here is, in this context, is that when a memory is retrieved or reactivated, when you use that memory or act upon this memory, then after you're finished, it stays as a stable long-term memory. However, the more recent revised model, which started with actually studies in experimental psychology in the 70s, but then received strong support from neuroscience experiments at the beginning of this of 2000. So according to the revised model, the short-term memory is the same, and even the process of consolidation from short to long-term memory follows the same model. However, importantly, potentially every time a memory is retrieved or reactivated, then it has to undergo an additional phase of consolidation in order to be maintained as a long-term memory trace in the brain. In extreme cases, as I will show in a minute, if the memory is not consolidated upon its retrieval, after its retrieval, it could be even completely degraded, as you can see in this illustration. Now, strong support to this revised model came from seminal studies done in Leduc's lab at NYU and spearheaded by Karim Nader the beginning of this century. And these studies are actually in animal models, specifically in rodents, using Pavlovian fear conditioning. So you have a conditioned stimulus, which is the tone, and the unconditioned stimulus, which is the shock. And when you pair the auditory tone with a foot shock, electric shock, then you create a trace of fear conditioning in the animal's brain. So in this experiment, they created this pairing, and after 24 hours, in which supposedly the new memory was consolidated, they reactivated the memory briefly just by introducing the auditory tone to the animal in the cage. And the results of this, and then they measured the behavioral measure for the fear memory is the percent of time that the animal freezes in its cage. Here you can see three groups. It doesn't matter at this stage because all of them underwent the same, didn't go any procedure at this stage. So we'll treat all of them as the same. And you can see that consistently, after this, when they tested the strength of the memory, the behavioral measure of percent freezing after this reactivation, immediately after, then you can see a clear signature of the fear memory trace when the animal freezes in its cage 70–80% of the time, relative to 20–30% of the time before the memory was created, which is the natural time probably that the animal freezes. It doesn't have to move all the time. So at this stage, there is nothing new, right? It's only the creating the fear memory and allowing its consolidation after 24 hours and obtaining a robust behavioral measure for this fear memory. However, after this brief reactivation, what they did is they injected protein synthesis inhibitors to the basolateral amygdala of the animal. And then they tested the strength of the memory 24 hours after this procedure. And these are two of the control groups, but the important result here is with this procedure, you can see the memory is virtually erased, completely degraded, at least its behavioral signature, you can see that freezing levels return to around 30%, which is which was the amount of freezing before the memory was ever created on day one. Additional experiments in this study show that this effect of memory degradation following its reactivation is specific to the reactivation. It's not only this procedure of inhibiting, injecting protein synthesis inhibitors to the amygdala, but if you do the same injections without reactivation, then there is almost no effect on memory strength. And there are additional specificities. For example, I mentioned a time window. So here you can see a demonstration of this time window of susceptibility following memory reactivation. Now you do the reactivation similar to the main study group, and the same injections as well. You just space 6 hours between the reactivation and the procedure, and the interfering procedure, and you can see that the memory is intact. Since then, there's been multiple studies in various animal systems, animal models, and memory domains. And the picture that emerged is that once an existing memory is reactivated, then a time window of susceptibility opens up for modulation of the memory trace. And when we started looking into this, evidence has been accumulating for similar processes in humans, mainly behavioral evidence. And the first question which we asked about a decade ago was, how does it operate? How can you test the operation of this mechanism in the human brain? So obviously we cannot inject protein synthesis inhibitors to the human brain. So what we used is non-invasive brain stimulation. So and specifically TMS, transcranial magnetic stimulation. In brief, what we do is we place a coil over the subject's head, and we induce an electric current through the coil, and by basic magnetic laws, then it creates a magnetic field which penetrates the scalp and the skull, and induces again an electric field affecting the neural tissue just beneath the surface of the coil. Now, I won't get into all the details, but I will just mention that there are various stimulation protocols. There is single pulse TMS, in which you can interfere with and this has high temporal resolution, and here you can interfere and establish a cause and effect link in specific cognitive tasks. And there's all these pattern protocols which were designed to mimic LTP or LTD mechanisms at the synaptic level. I will focus here on one of these patterned protocols, which is R stands for repetitive, so it's a frequency of 1 Hertz repetitive TMS. And in the motor domain, what we can do is we can, all this field of research using TMS originated from studying the motor domain, because here you can get clear physiological measures together with the stimulation. So if you stimulate the primary motor cortex, the signal flows through the corticospinal tract and then activates the muscle from which you can connect electrodes and record the MEP, or the motor evoked potential. And we can also track, we can define the target for stimulation based on anatomical or functional brain imaging, which we do before the experiments. And we can track through a neural navigation system, we can track the stimulating coil as the session progresses. Now, what I started to say with regards to the MEP and the 1 Hertz stimulation protocol, this protocol was shown to be a protocol that inhibits corticospinal excitability, and that has been shown in various studies. As you can see here, this is the MEP, the amplitude of the MEP recorded from the FDI muscle here in the hand. And you can see that before the repetitive 1 Hertz inhibitory protocol, the amplitude is marked here, and you can see a decrease in the amplitude of the MEP following the inhibitory 1 Hertz stimulation, pointing to 1 Hertz stimulation being a protocol that inhibits corticospinal, cortical brain excitability. And this has been replicated also in the visual domain using measures of excitability, signatures of excitability in the form of visual phosphines. So now you may better understand the rationale that we had in mind, replacing the inhibition with inhibitory TMS paired with the reactivation, in this case, of a motor skill memory. And what we showed is that behaviorally, we impair the performance of the subjects measured a day after stimulation. And also using resting state fMRI, we showed degradation of within the motor network here, connectivity at rest between the sensory motor striatum and the primary motor cortex, which was impaired following the active stimulation relative to control stimulation. And of course, subjects can regain this connectivity easily if they train again with the motor skill test. So after this long background, I hope you now better appreciate the rationale we had in mind for applying these brief reactivation to experiments in perceptual learning. Now here, what we had in mind is if this reactivation opens up a time window for additional consolidation or reconsolidation of the learned information, then here we won't induce any interference, not by injecting anything, neither by TMS, but just have these brief reactivations of only several trials. And if this story of memory reactivation is reliable, then maybe these brief reactivations will enable efficient consolidation and induce replication of this full learning curve, replacing these hundreds or thousands of trials. By the way, you can stop me at any time if you have any questions. So this was our first basic study using this approach. So we used the texture discrimination test developed by Carney and Sagi. It's a well-studied model task for evaluating effects of perceptual learning. We had two main groups here: the standard full practice group, which practiced with hundreds and thousands of trials over 5 different sessions, and the rapid learning or reactivations group, which first encoded the memory with an intense practice session, and I will get to that later. The rationale is that in order for reactivating a memory, you need to create it first. So first we reasoned that we need an intense session on day 1. And then these brief reactivations of only 5 trials per session. And this is very different from any perceptual learning experiment. So the participant comes to the lab, goes to this small room, adjusts to the dark and everything that you do in perceptual learning studies. But after 10 seconds and 5 trials, he's out of the room. And then we do a final retest on day 5. And the basic results show that here in blue is the replication of the well-known between-day learning curve, the standard learning procedure. And you can see in the rapid learning that subjects get to more or less obtain similar thresholds without extensive practice with these brief reactivations. And since then, we've also showed similar evidence, and we went back to motor skill learning, and I will get to that later on in the talk. Just to expand a bit about this study, so we use the texture discrimination task, as I said, developed by Carney and Sagi. And here the task is to, while you focus on the center of the screen, and there is a letter fixation task, a T or an L, but this is just to enforce fixation. And in the periphery of the visual field, you have to discriminate whether these three diagonal lines relative to the horizontal background are in a global vertical array or horizontal array. Shortly after the target frame appears, we introduced a visual mask that limits visual processing. And as we decrease the temporal distance between target and mask onset, then the task becomes more and more difficult. And these are the original results from Carney and Sagi with this task. You can see here the psychophysical learning curves. The subjects come on day 1. They're at a relatively high SOA, near 100% correct. And as the SOAs decrease, then performance gets to more or less chance level. And you can see that with additional training, each psychometric curve here is a different session on a different day. You can see this leftward shift of the psychometric curve, indicating offline learning in this task. And you can extract the visual, the discrimination thresholds from each of these curves. Here, each threshold was measured as 80% correct. And once you extract these thresholds, you obtain the between day learning curve, showing that clearly that visual thresholds improve between days. So what Rotem and Ronnie did in the lab is use exactly the same task and the design I mentioned earlier. And I spoke about this main result. And we showed that reactivation is both sufficient and necessary. Without reactivation, there's hardly any learning. Initially, our rationale was to design the reactivation trials, these 5 trials, to adapt them to each subject based on their threshold measured and derived from the session on day 1. But then in an additional group of subjects, we showed that this operates also in far threshold conditions of the reactivation. We showed that this reactivation is long-term and was retained for several months. We did a lot of control experiments. I will introduce just the 2 main controls in my view. The first is we asked whether this may be just an epiphenomenon of retrieval enhancement, some strange priming effect or something of that sort. So to test this alternative hypothesis, what we did is we reasoned if this is the case, then what we need to do is to put these 3 reactivations one after the other and just before the retest on the second day. But as you can see here, when we plot the retest against the test thresholds and also in the averages, you can see that there's hardly any boost of learning here. An additional control that, and this may indicate also that this may indicate that the reactivations also need to be on separate days, maybe because sleep is an important factor here, playing a role in each phase and cycle of reactivation, reconsolidation. And this is something we're looking into at the moment in follow-up studies. And the final control I will show you for this study is, could this be just some kind of magic of that in our lab somehow, in contrast to multiple studies in the field, we're able to improve human perceptual thresholds just by a few trials. So from day 1 in this condition, from day 1, participants performed only a few trials. and then they were tested to evaluate their perceptual thresholds on the final day of the experiments. And as you can see, this did not result in reduced thresholds, pointing to the, as I mentioned before, to the notion that you probably need an extensive session on day one in order to create a memory of the skill, and then you can start talking about reactivating it. So this is, according to our interpretation, why this did not work in this condition. So to conclude this part, we show that brief reactivations of consolidated perceptual memories enable perceptual learning. That this reactivation-induced learning was not a manifestation of temporarily enhanced retrieval and was retained for months, and that short training per se did not result in reduced perceptual thresholds, pointing to the importance of initial encoding. And this may lay the foundations to develop strategies to optimize learning in normal and clinical conditions. And I will get to that point also a bit later in the talk. As I said, we also have now similar evidence, and this is study spearheaded by Jasmine, a PhD student in the lab, in collaboration with Hagai Sharon from Ichilov. And here we applied a very similar design in the motor skill learning domain. So in brief, participants have to perform a repeated sequence of finger movements using this response box. And we had a few groups here of reactivation, of full training, and of just a control group of test retest. And the main results show that also here reactivations were very efficient in inducing motor skill learning. And furthermore, we identified here that they are not always efficient. And actually, it depends on, in this case of motor skill learning, we show that it depends on the quality of the brief reactivations. And one thought would be that you need to have the reactivations intact, meaning performance of the sequences without any errors or with minimal amount of errors. But we show that this is not the case. And what determines whether a reactivation is efficient or not is not merely whether the memory is correctly reactivated, but what I show here is the continuity score. So you need a few sequences in a row, error-free, in order for this reactivation to be efficient. So this is just an example of two different reactivations of a sequence of 4, 1, 3 to 4. There is exactly the same number of errors here or correct sequences in the two cases, but here you can see that they're more spread out, they're more interleaved here, the errors, whereas here there's a continuous sequence of movement without any errors. And this is an example of where reactivations were efficient, and this is an example where they were less efficient.
Nitzan, can I ask something?
Sure.
Yeah, I want to ask, I mean, what I see here, I mean, the sequence and whether you do or you don't make errors and how they are distributed within the sequences is in a way dependent on the performance of the participant, him or herself. So it's not that you, the experimenter, can control it. So what do you do if there is no efficient sequence or smooth, continuous, non-error-free sequence?
Right. So this study was just to basically understand the mechanism. And now we have follow-up studies in which we try to forcefully induce reactivations which are error-free. So you can do that. You can do that with reward, or you can do that by clamping the speed of the finger movements so that they do it with in a low speed without any errors, but this has also challenges in itself. So we're in a process now of trying to, in experiments, trying to have the subjects do the reactivations with a high continuity score.
Okay, thank you.
This is a big challenge. We showed here that the behavioral measures were also correlated with measures of corticospinal excitability measured over by stimulation of TMS over M1. And in a follow-up study, we show that this does not operate at a micro time scale. So it's very similar to what I showed you in the perceptual domain. So we show that this needs to be between days, this whole notion of reactivation, and it doesn't operate if you do these reactivations within the same day. At least does not induce significant learning, as you can see here. So following these basic behavioral studies, we started the quest to try to understand two main things. The first, in this big, what we call rapid learning brain project, which is ERC funded. So first is to understand the core mechanisms of this learning mechanism and the role of sleep, as I mentioned before. And secondly, to try and see whether this can generalize to additional memory domains. And a subportion of this aim is to see whether this also operates when memories are unwanted and not only when memories are beneficial for learning. And I will get to that towards the end of my talk. Dean and Tali from the lab, they started to look at the neural mechanisms underlying this form of learning. And our working hypothesis is that the systems level mechanisms underlying this form of learning involve engagement of higher order regions, which then interact with the early visual regions. It's not that higher level regions are not involved in the standard extensive practice-based perceptual learning, but there is a very dominant contribution of the lower level regions, such as primary visual cortex. And this makes sense, right? Because when you want to induce plasticity related to very basic perceptual visual tasks, like low-level perceptual visual tasks, it makes sense that you need to bombard the very hardwired early visual system in order to induce some form of plasticity there. So this is the basis for our rationale that in the case of reactivations, maybe we can bypass this bombardment by engaging in a more prominent manner by higher level regions, which then in turn interact with the early visual regions. And the second part of this story is to try and to causally modulate learning, similarly to what we did in the motor domain. And here we have preliminary evidence showing that if we stimulate, in this case, primary visual cortex paired with the reactivation of the memory, then we're able to some extent to degrade the learning curve. So the big project going on now, spearheaded by Tali, a PhD student in the lab, is to replicate this the experimental design you've seen before in the scanner. So we have here two main groups, the full practice group and the reactivations group. And they undergo anatomical and functional MRI before and after learning, just before the final retest. And in the scanner, we have the texture discrimination task and a control task in which we try to maintain as similar as possible visual stimulus, but a very different task. So here they have to do only the fixation, and there's actually no task to perform here in the periphery. And these are very, very preliminary results, really just came from the oven. And we can see here that following learning, when we contrast the reactivation with condition with the full practice condition, you can see enhanced activity in attention, higher order attention-related regions along the dorsal attention network. Another story here, which we're trying still to understand, and it's still very preliminary, is the other direction with the default mode network. So this is all very preliminary, but it does provide some indication that indeed this form of learning is somewhat different, although the results, the behavioral results are the same, meaning the subjects arrive to the same level of performance after reactivation and after full practice. It provides some indications that the brain mechanisms are very different. Another line of studies is, well, if this reactivation is an opportunity to modulate the memory trace, then this could be very important and efficient in terms of generalization of learning. So a big story, as you know, in visual perceptual learning, but also in skill learning, is generalization. When learning is often, there's this chains of specificity that you're locked to this specificity, whether it's specificity, retinotopic specificity in perceptual learning, or in motor skill learning, specificity to the learned sequence or to the trained hand. What we try to do is to leverage this reactivation window and induce during this reactivation window also signals that are related to generalization. So brief introduction of the sequence with untrained hand or untrained visual location, and to test whether this can enhance generalization patterns and unlock the very well-known specificity in these forms of learning. And I mentioned interactions with other learning domains. So so we try to test these interactions with the hypothesis that if we synchronize reactivation in one domain with new learning in another domain, then maybe we can also enhance learning. And this is, I think, a very nice study that Shira did in the lab. So what she did here, and the two domains here are the motor domain, specifically the oculomotor eye movements, and the perceptual learning domain using the textual discrimination task. So first, what Shira did is develop a task in which participants using eye tracking are trained to perform a specific saccade to in a specific vector, amplitude and angle, and to try to make this task as oculomotor as possible, meaning we remove the visual cues very early on, and they're, so to speak, they're blind in the room, and they only get an auditory cue indicating that they perform the right movement, the movement that we want them to learn. This is a very, very difficult task and very painful task. But after a lot of training, they're able to learn. Again, they learn to make an eye movement. They have no visual feedback. That the only thing is that they see is they have to fixate randomly the target that they start from appears randomly on the screen. It changes from trial to trial, which makes the task even more difficult. And they have to fixate on the starting point, and then it disappears, and they have to make an eye movement. They have no visual cue at all, and they're able to learn this this vector of movement. And the experimental design here is that after this oculomotor learning, then they are briefly exposed by brief reactivation of this task. And this is paired with new learning of the textual discrimination task that we've seen before. And on day 3, they're retested on the visual textual discrimination task, the TDT. And you can see here that these are the learning gains in the group that did not undergo the oculomotor learning and reactivation. And it's in the order of 5 or 10 milliseconds. This is something expected over 24 hours. But here you can see how this procedure boosted learning in this is in the visual domain. This is the learning, the offline learning in the textual discrimination task using this pairing of oculomotor reactivation and the visual task. This pairing was not also temporal pairing. We also made sure in the design of the experiment that this vector is exactly the vector of where the texture task is relative to the fixation. However, importantly, in the textual discrimination task, remember, they're not allowed and they're not able to move their eyes because they have to fixate at the center of the screen. And when we retested again the oculomotor performance, we showed also this is only a correlation, but some correlation evidence of pairing between these two tasks, because there was a high correlation between the visual learning and the oculomotor gains. We're not trying to understand also how this operates using brain imaging. And more importantly, in my view, we also started to look at the causal mechanisms. And here we leverage a procedure of closed loop TMS EEG. So we record online EEG activity from the visual cortex, and based on these recordings, we apply in a phase-locked manner single pulses of TMS over the frontal regions, in this case, the frontal eye fields. And the goal is to try to facilitate this hypothesized connectivity between the early visual regions and the higher order regions. And this project is in collaboration with Tübingen, Ulf and Christoph, and Shlomit, our neighbor here, Shlomit Yuval Greenberg. Again, just to illustrate what this is also spearheaded by Shira from the lab. So as you know, the visual information flows first through the thalamus to the early visual regions, and then in a feed-forward manner to the frontal regions, and then there's this feedback loop, of course. And what we try to do is to increase, artificially increase this connectivity between the occipital and the frontal regions. So as I said, we record EEG activity. Specifically, we're interested in the alpha frequency band, and once it elevates beyond some thresholds of alpha power, then we calculate the phase of the alpha activity, and we project when it should arrive to the frontal cortex. And here we stimulate using the single pulse TMS the frontal eye fields, and of course, after our stimulation, the signal flows in a feedback manner back to the visual cortex, which is why this is a closed loop. And in this design, and this is also very preliminary data. So we have the stimulation group, and they do a basic visual detection task. And in between, we do the closed loop EEG TMS. And we have here a pilot experiment with a no stimulation group. We still need to collect real control data with real stimulation and sophisticated control that we usually do in our experiments. But at this stage, it's just a no stimulation group. And very preliminary data shows that indeed visual sensitivity was enhanced between sessions, and only in the stimulation group. And as expected, it did not change in the no stimulation group. This was done immediately after a short break. So this study is really a beginning of a new avenue. And at this stage, as you can see, it's only about short-term effects, but we hope to extend this design later on to offline between day learning as well. And the last part of my talk, as I promised, we remember in the title there's in parentheses unlearning, and I mentioned that we aim to extend this this view also in relation to negative events or negative memories. And the rationale here is that if this mechanism operates in the brain, then the brain doesn't know whether it's a good memory, so to speak, beneficial for learning, or a maladaptive memory in the form of a memory, a negative memory following a stressful event. And the hypothesis here is that in the case of maladaptive memories, what happens here is that, and I would just say I'm not a clinician, but and this collaboration was done by a joint postdoc, Noah Hertz, in collaboration with Yair Bar Chaim's lab. But I can tell you that in PTSD, one of the unsolved hallmarks of PTSD is intrusive memories, which are memories that are encoded as part of the trauma and then come as kind of flashbacks in day-to-day life. And in extreme cases, really, people can hardly function in their daily life when these intrusive memories become very, very maladaptive. And what we thought here is maybe these visual intrusive memories form as or play a role of spontaneous reactivations. They're not reactivations done in the lab, but they occur in real life in a maladaptive manner. And this is why this memory, if reactivation, as we showed in our basic study, that reactivation strengthened the memory trace, then maybe this is the case also in the case of maladaptive memories, of negative memories, that these intrusions, repeated intrusions, serve as repeated reactivation, which strengthen, in this case, a maladaptive negative memory. And the first step to test this hypothesis before moving on to a big study in patients that we are starting these days is a study in healthy volunteers. Fortunately for them, they don't have a trauma, they're healthy volunteers. And we create a mini trauma in the lab. And this is not something we invented. It's the trauma film paradigm, which is a sequence of short, very unpleasant video clips, is something which is highly used in many labs in the world. So we took this well-known paradigm, and we first kind of induced the model. Again, this is just with all limitations, this is just a model for a trauma. And immediately after encoding, they underwent a visual recognition test. I will show you more details about this in a minute. And after that, they went about their usual daily routine. And every time an intrusive memory came to mind, they recorded it in their in a designated app in their phone. After 5 days, they returned to the lab for an additional visual memory test. And I will show you, I will talk more about the main results in a second. I just want to expand a bit about the study design. So as I said, they first encoded the trauma, and there are 8 scenes, from unpleasant scenes in this experiment. And then after the experiment, they go about to their usually daily routine, and they record these intrusions. So each color here is a different scene. And on day 5, they return to the lab for a visual recognition memory test. And this is a type of the visual test. It's a 2AFC, very basic visual recognition task. Then, as I said, they record in their online app, and then they come for the memory test again. Now, after the experiment is over, we can separate the scenes for each subject to scenes that have intruded to memory and to scenes that did not intrude. Okay, and this is an important point because we can do it for each specific subjects, and after that we don't care anymore about the scenes themselves. So scene 1 could be intrusive for subject X and not intrusive for subject Y. But after we separate intrusive and non-intrusive, we don't care about that anymore. So we have just over the entire group, we have for each subject intrusive scenes and non-intrusive scenes. So we can do this separation, and this is after the experiment. And we also can look back at the results of the recognition test on day 1 and separate the results according to, analyze the results according to the, at this stage, I would call it maybe more accurately the to be intruded scenes, right? Because they did not intrude at this stage immediately after the trauma fields, but these are the to be intruded scenes and the not intruded scenes. And these are the results. So I will start from the results after the experiment. So you can see that by the end of the experiments, you can see a clear difference between the visual performance in the intruded scenes relative to the non-intruded scenes. So the first question you can ask is, so what, right? Because it's expected, right? Because if these are intrusive memories, and they're probably strong memories, so they remember them better, right? So it's not very surprising. But I think an important point here is to look back at test 1, which is what I mentioned earlier, and you can see that immediately after encoding, there was no advantage, so to speak, of the intruded scenes over the non-intruded scenes. If any difference, then it's a difference actually in the opposite direction, but definitely not what you see by the end of this of the experiment. This indicates that the development of the memory occurred together with these what we call spontaneous reactivations and what all the clinician call intrusive memories. We proceeded then to an initial study also at this stage in healthy volunteers to look a bit into the brain. And what we did is first you see here results of resting state fMRI, and we parceled the brain into different ROIs, and you get this matrix. And what we observe is that the connectivity between visual cortex and the amygdala was the best predictor for the emotional intensity of the intrusions. These are very preliminary results, and I have to say again, this is in healthy volunteers. What we proceeded to do then is to stimulate the visual cortex with inhibitory RTMS, 1 Hertz inhibitory RTMS, immediately after the reactivation of the memory. So in this study, reactivation was, the first reactivation was not spontaneous, was in the lab. So they underwent a brief reminder protocol, and immediately after this reactivation, we stimulated the primary visual cortex relative to control vertex stimulation. And what we see is that this stimulation reduced the intensity, the emotional intensity of the intrusive memories recorded then in the online app. But it's important to say it did not affect the frequency of the intrusions, only their emotional intensity. And now what we're doing in the lab is totally different experiment in terms of the stimulation protocol, but the theoretical framework of the reactivation and inhibition is the same, but with PTSD patients. And again, this is we've just started a pilot in this line of work. Current directions include what I mentioned before, answering your question, Sharon, of how to personalize this thing of in a designed manner to personalize this learning. So Dekel, a postdoc in the lab, together with Yasmin, they're leveraging machine learning approaches to try and identify features in, and this is in motor skill learning, to try and identify the features that are the best predictors of the degree to which participants benefit from reactivation-induced learning. So it's a much more sophisticated analysis approach compared to the preliminary analysis that I showed you before of the continuity measure. And the goal is, once these features are identified, then to try and adapt them for each participant according to their initial performance in the task. Another line of work or from the lab. So far we spoke about one task in a minimal amount of time, but if this works, then maybe we can also speak about several tasks in the same amount of time. This is a very sophisticated, it looks simple, but it's a very sophisticated design and approach, because we have to make sure that these reactivations done one after the other are not creating interference between the different tasks. So this is a very long journey in terms of looking at parallel rapid learning. And finally, I will end with the implementation. So we're looking at ways to implement this for daily life. And Gilad from the lab has been looking at the multiplication table together with Dror Dotan from the education here faculty. And we're looking at adult healthy participants, so not at children. So they already know the multiplication, of course, most of them. So Gilad designed some new language of arithmetics that kind of mimics the multiplication table, and to test whether this can be learned more efficiently with the reactivation methods. I mentioned the PTSD study that we're starting now, and in the next slide, I will show you some data in relation to autism. And finally, Gilad is also developing virtual reality platforms to see whether we can make the reactivations more vivid in additional memory domains, and to try to enhance learning using the combination of what virtual reality can provide you with the theoretical framework of memory reactivation. And I said I would just mention this study of, this is in collaboration with Ilan Dinstein from Ben-Gurion University. And here what Shira did is applied the reactivation in perceptual learning, the reactivation-induced learning in a high-functioning ASD subjects. And from these results, what you can see is that reactivation successfully induces learning, but also enhances generalization in high-functioning ASD subjects. And this is a really interesting result, because one of the challenges in autism spectrum disorder is they don't necessarily learn in a less efficient way than neurotypicals. And I must say that in some domains of learning, they're even better, and sometimes much better. But what is a challenge there many times is that learning is locked to the notion of specificity, and it generalizes in a less efficient way compared to neurotypicals. And what we show here, and this is a behavioral study, but it's in line with our general hypothesis of the engagement of higher order brain regions in this form of learning. And we show here increased generalization in ASD following reactivation-induced learning. So to sum up, we propose to rethink the notion of practice makes perfect. We're not saying that it should be exterminated. I still, of course, I think that many learning domains, you cannot replace practice. But we propose that in some, and our goal now is to try and identify which are these some and which are not. In some domains of learning, we propose a more economic form of rapid learning based on the reactivation framework. And the ultimate vision is that we can impact really daily life function as we're slowly starting to test carefully in the lab. So I'd like to thank, first of all, you for patiently listening to the talk, and my lab members, collaborators, and funding. Thank you very much.
Thank you, Nitzan, for a wonderful and very exciting talk. And I want to invite everybody to unmute yourselves, and let's give Nitzan a big applaud. And I want to open the floor for questions, but I'll start. So I have a lot of questions, but I'll start with one or two of them. So first of all, when you showed the learning of making a saccade in the dark, and then the fact that it actually enhances the perceptual learning when the vector of movement was toward the area where the saccade was actually, where the target was. So I wanted to ask, and you tested it on on the perceptual learning paradigm, but potentially maybe maybe you have enhanced pre-saccadic attention mechanisms, and maybe this can generalize to additional tasks in that location. So have you tested other tasks, not ones that have been learned? So maybe you have, I'm just suggesting, I'm talking about the psychophysics. Yeah, that's my first question. Shall I continue?
Yeah, I can briefly answer. Yeah, so we did not test the generalization to other tasks. We have tested the generalization to different vectors, and it was specific to the learned movement. And what you're saying about attention, it may be an explanation. I think it fits well with our thought that regions such as the dorsal attention network may be more engaged in this form of learning. So this very well be one of the mechanisms. Yeah.
Okay, wonderful. And another question that I have about when you tested the learning and the intruded versus to be intruded, or and there was no difference upon just after the exposure, just following the reports and everything, you found difference. And so I was wondering, I mean, what you showed was perceptual or behavioral measures, but I'm wondering if maybe there were already differences, maybe in skin galvanization response, heartbeat, physiological measures, or maybe even in that session to ask, just a little bit of elaboration, free elaboration, to see maybe they not only they should rate, I don't know how intense, something about the experience maybe could even be predictive at that stage. But anyway, beautiful result.
Yeah. And these are good ideas, and we're implementing a more rigorous and complete behavioral design in terms of the clinical behavioral measures now when we're doing the big experiment in patients.
Okay, great. Good luck. Yes. Additional questions? Okay, I have more.
Okay.
About the DMN, well, it's not a question, but you said that you're still trying to understand why, if I understand correctly, it is more deactivated in your reactivation paradigm. So I just want to say that the DMN is more activated when there is more resources, attention or external resources directed toward the external world, let's say. So it could be that this is basically an indirect measure of the amount of resources. I mean, my assumption is that if you examine the amount or the magnitude of the modulation, you will find that it's in the same magnitude as the positive activations that you get. I can discuss it with you further, but
yeah, we will. Yeah, it's an interesting point. Yeah, we're trying to understand this result. Yeah.
Yeah, so it could very well be. Okay, I'll let others. Okay, actually, I have more things, but
See that someone is raising his
yes,
hand.
Joseph, do you want to ask?
Yeah, that would be great. Fabulous talk. I really like the conceptual idea of rapid learning.
Joseph, your audio is not so well, so maybe you
or
would,
almost
maybe...
like one trial learning. And we do some work in so let me go back. So the interesting part,
maybe you can turn off your video so the audio will be heard better. Yeah.
Can you hear it now? Great. Is that better?
Yeah, it is.
That might be better. Yeah, I'm at home in the rural country of outside Toronto. So what is interesting is your rapid paradigm where you pull the signal from one source in visual cortex, use the alpha power and put it back into FEF and see interesting modifications that seem to be learned, right? What I'm curious about is why you would pick the alpha rhythm, and were the people's eyes open and closed when they were doing the EEG?
Right. So the eyes were open, and yeah, the rationale for the alpha is just because it's, as you probably know, it's a signal that, you know, it's a well-known form of communication between attention-related regions and the visual cortex. So that was the rationale.
Okay, great. Yeah. So do you know of any endogenous signal that you know that has been measured by EEG and other paradigms or something similar within the saccade or attentional paradigm where a person learns something and the alpha rhythm or alpha peak or alpha power got increased?
Yeah, so I think there may be some work there, but I just want to highlight that we are looking at this stage of the experiments in terms of the closed loop. We're looking at actually not at learning. So they're not because the default group here does not learn. It's sessions that are immediately one after the other. So this experiment is different from what you've seen in the rest of the talk of between session learning. So this is just really a very kind of brief enhancement of communication. People have been doing.
Okay, so how I might have sit in the. Sorry, I might have missed it. How long was this closed loop intervention with the TMS and the EEG?
It was just several minutes. And the task was performed immediately before and immediately after. So it's different from the between day thing that I've referred to in the rest of the talk, because it's really just the beginning of this line of work.
Yeah. Do you think So do you imagine a case where a person for that 2 minutes, instead of doing the TMS as a control paradigm, they like ran on a treadmill or something like that?
In
Would
order
that be analogous in terms of like a 2-minute intervention?
So what is the rationale for having them run on a?
So exercise, I think exercise and some other paradigms where people are doing something shows improvements in brain processing, right?
So I think, yeah.
So that would be like an endogenous
Yeah.
control in some way.
Yeah, could be interesting. I think so. I think if I understand you correctly, it's not really a control. It's another research question. So it's
it's a proof of concept, but it's
right.
more proving that the mechanism that you're playing with is a mechanism that is endogenous within some pillar of research that exists, right?
Interesting.
Because what we do in our group, one of the small lines of research is playing with dance, and people with Parkinson's dance, and their disease slows down. They get better tremor, better balance, better gait, better quality of life, better mood. We published all this work just in the last few years, but we measured EEG pre-post dance class. So it's not 2 minutes, it's an hour. And we see increases in power in kind of frontal polar like area would be close to like SCF, supplementary motor area, and in the back of the brain. And we see increase in alpha frequency in the Parkinson's people and increase in alpha power. But it's an hour, and they have a disease. So it's not an intact brain. So
interesting.
That's why I asked about the exercise. Yeah. Nice. All right. Thanks for your question. Thanks for your studies. It's great. I'm going to follow some of your work now. It's awesome. Thanks.
Thank you.
Nitzan, Eliran has asked, and he thought he may not be here to hear the question, the answer. So he asked in the chat, does this approach that you're proposing, can it be used to interfere and heal bad memories in post-traumatic patients? I think maybe you have started to answer that later. I mean, it sounds like
we started to ask that, we're far from answering it, but we started to ask it in the biggest experiment. Yeah.
Okay. So I will inform him about that. And I wanted to ask about the procedural learning, following what Joseph has suggested. When you do the motor or the procedural sequence, is it coupled with music or some, I don't know, auditory rhythm that may assist? Or is it just I don't know, how long is it, by the way?
The motor skill learning of tapping a sequence, the standard task is 12 to 15 minutes that they repeatedly tap the same sequence over and over again. And the reactivation that we did is only 30 seconds, one trial. Each trial there is 30 seconds, so it's one trial of 30 seconds.
I see.
And there's no music. They're just instructed to tap as quickly and accurately as they can the sequence during a limited time frame that they have.
I see. Okay, interesting. I'll discuss a lot of ideas with later on with you. I'm sure some of them you'll discard immediately, but maybe something will be helpful.
Sure.
Okay, so if there are no more questions, then we will stop here. And I want to thank you again for a beautiful talk. Very inspiring, and I hope leads to many additional breakthroughs. And I also want to say that next week, since there's VSS, we will not have a talk, but in 2 weeks, Ro'i Salomon will be talking about, on the 24th of May, is the location, about virtual hallucinations, studying the sense of reality with virtual reality. So we will stop here, and thank you so much, Nitzan, for joining us.
Thank you very much.
Yeah.
Okay. See you soon.
See you. Yes, definitely.
Bye-bye.
Bye-bye.